above ground portion. But I also want to enlighten you that the root system on some plants is way bigger than what you see above ground. So even if you see, a pretty substantial tree, that it depends on the species, but very often the root biomass is more than what you see above ground. And so the below ground happenings for the plant body can be quite substantial, and they can be putting in a lot of effort to make sure that their root system is robust and big and constantly growing. So now we need to think a little bit more. I'm going to kind of zoom in to the cell area and think about what makes plants kind of unique. So the cell wall, we don't have cell walls, right? We just have a cell membrane. Plants have actually two cell walls, at least some of them have some of their cells. And really, when you start thinking about the cell wall and how strong it can be, that helps to explain how they can stay upright without any kind of skeletal system. Like we have a nice skeletal system that we can hang all of our organs and skin on, and it helps us fight gravity, stay upright. They don't have that. So they have to deal with, other strategies. One way, they have more than one. One way is to have a cell wall around they have a primary cell wall around all of their cells. And then some cells have an additional cell wall. So really, this is one of their big ways to have some structural support, fight gravity. So we talk about a primary cell wall and a secondary cell wall. And I'll go into a little bit more detail on each of these. But a primary, so anytime when that seed starts to germinate and grow, and you have cell division going on, each new cell will have a primary cell wall. All cells will have a primary cell wall. So it's relatively thin, and it's, all living cells, definitely, they have a primary cell wall. Secondary cell wall, not all cells of the plant necessarily have a secondary cell wall. If they do have a secondary cell wall, it'll be thicker than the primary. It'll be stronger than the primary. And it is kicked into production once the cell is maturing, or pretty much fully mature. Then it'll get the signal, okay, we're destined to have a secondary cell wall, start making it. All right, so it's kind of a slow or a delayed process. Every cell in a plant body has a primary cell wall. Only some cells have a secondary cell wall. And then they don't really start manufacturing it until that cell is pretty much mature, it's reached its full size, it's got everything going on inside, and then it'll form a secondary cell wall. Plants are multicellular, just like us. They have to have a way for all of these cells to stick together, for lack of a better way to describe it. So the middle lamella, they often use the term cements. It cements the cells together into the different plant tissues that we'll talk about. And a big part of the middle lamella that's allowing for all of this cohesion of the cells is pectin. So if you've done any making of jams and jellies, that's what you're working with. It's actually extracted from the tissues of plants. And then they sell it in the store for you to use as a thickening agent. It helps make your jams and jellies thicker. And so it does have that gelling consistency. So if you have any experience with that, it's you kind of see how that can be used for the neighboring cells to be able to stick together. Here's a mock-up of some neighboring cells. So we see the primary cell wall is the thinner, darker brown cell wall. And then you can see that this one, these cells anyways up in the foreground, they have a secondary cell wall. And you can see how the secondary cell wall is thicker than the primary. And it's also laid down to the inside of the primary. That, when I was first learning that seemed weird to me, but that's how they do it. Instead of having it on the very, very outside, they stick it to the inside of the primary cell wall. And you can see, we'll go over a little bit more what makes up the primary and secondary, but you can see the thickness difference, at least in this one. A couple of other things that I'll be pointing out in a little bit here. Well, here you see the middle lamella. They're trying to show how that's the glue or the cement that holds the cells together. And then you can see the artist drew these, I call them tunnels between cells. You have to imagine, they've cut away the cells, but you have to imagine this being an entire cell. And then there's tunnels that can connect neighboring cells. And we'll talk more about those too. And one other thing to point out on this drawing, this cell in the background here, this big kind of whitish blob, and then this purpley blob. Does that remind you, does the purpley blob remind you of an organelle that you probably learned a lot about in other classes? Yeah, nucleus. And so at least, I don't know if they still teach it this way, but when I was learning cell structure, animal cell structure, they always said the nucleus is the biggest organelle. And that's true in the animal world, but they make it sound like it's universal. All cells are that way, and it's not in the plant world. So in the plant world, when you have a mature plant cell, the nucleus is not the biggest organelle. It's actually this thing, which is called a vacuole. And we'll talk more about the vacuole. But I wanted to put this visual in your head now to impress on you, or impress upon you that it's big. And when a mature cell is manufactured, they call it a central vacuole. This huge blob, and there's lots of stuff in here, but it takes up most of the volume of the mature plant cell. So we'll come back to that as well. But let me finish the thought on the primary versus secondary. So very often when we denote primary, the word primary, we use a 1 with a degree symbol. That means primary. So the primary cell walls, when you look at their composition, there is pectin in the primary cell wall, cellulose, hemicellulose, and protein. You don't need to know the percentages. I put them in there to, if you're interested, but I won't be asking about this. However, the actual composition of what's in there, the words. We'll look at the words here. And I'm going to get a little bit more into cellulose because that is a very important part of the cell walls, both the primary and the secondary. And it's kind of a weird thing because when you look at cellulose at the very basic level, it's just glucose units. It's sugar, which, some of you dissolve sugar in your coffee. It just dissolves. How can that be strong? And so, but it is. The cellulose is incredibly strong. And so what we see is that it's glucose. So the very bottom of the image are a bunch of glucose molecules bonded together into long strands or chains. And we call that cellulose. Okay, but really the very, very basic unit of cellulose is just glucose. Bring them together, then you have what we call cellulose. Now what the artist is trying to show here is that you have a cluster of a bunch of chains of cellulose all clustered together. So that's what this is showing here in the microfibril. So they cut it in cross section and saying each one of those little whitish things is a chain of cellulose. Does that make sense? Okay, so they're all kind of clustered together. And we call that a microfibril when they're a bunch of cellulose chains or strands are clustered. Then you take a bunch of microfibrils and cluster those together. So that's what a fibril is. A fibril is microfibrils clustered together, and the microfibrils are a cluster of cellulose strands. So it's this kind of a nesting arrangement, and the way things are clustered together, it really does provide a lot of structural rigidity and support, even though when you come way down to it, it's just glucose. But the way it's put together in 3 dimensions into the microfibrils and fibrils really makes the difference. I often use the analogy when you're thinking about a cell wall of a plant that it's like reinforced concrete. If you've ever been to a construction site where they are putting down the steel rebars and then the cement mixer truck comes along and pours the cement across the rebar, and then it solidifies up into concrete. So that's kind of what this image reminds me of, where you have the cellulose and hemicellulose as kind of like the rebar structures, steel rebar, and then the pectin and some of the proteins, that's kind of like the cement that's being poured across it all. So when you have that kind of structure, it really does, it's not just the cellulose, it's everything, but certainly cellulose is a major strengthening aspect of cell wall, but you certainly have the pectin and the proteins and the hemicellulose all playing a part. Okay, now the secondary cell wall there's some similarities, but in that image I showed you, it's thicker typically than the primary. So there's more cellulose. They've beefed up the cellulose content. There's actually less pectin, probably to make room for the cellulose. And it has an additional chemical that you don't find in the primary. So this is lignin. And lignin is even stronger than cellulose. So if you see some plant cells that have a secondary cell wall, they are super, the structural support is just great because of the lignin. They didn't actually know the chemical composition of lignin until fairly recently because it's so complicated. There's all these side branches and it's complicated. And so it took a long time to piece that together. Lignin is the reason, if you've ever worked or driven by a paper producing plant that makes, paper products, and you might smell some weird smells. Those are the chemicals, some of that smell are the chemicals that they use to essentially degrade the lignin and try to get most of that out, because good quality paper has low lignin content. And so when you take the tree trunks that has a lot of lignin, you got to kind of strip that out. And lignin is so chemically complex that they have to attack it with some pretty harsh chemicals to get that to reduce the content of lignin in the final paper product. Definitely more dense than a primary cell wall. And like I said and showed you in that picture, when a plant cell decides, okay, I need to put down a secondary cell wall, they'll put it down to the inside of the primary cell wall. And it's formed after the cell is done growing. So once the cell has reached its mature size and it's good to go, if it's destined to have a secondary cell wall, then the secondary cell wall production will kick in. And the reason that it has to happen that way is that once the secondary cell wall is formed, believe it or not, that cell then dies. The internal contents of that cell die off and go away. A lot of it probably gets siphoned off to neighboring cells and gets recycled, but basically that cell will die and it'll end up to be a hollow area. There's like no cytoplasm, nothing. And that may sound weird that they deliberately do that, but actually, if you do it in strategic areas of the plant body, it's actually a great idea, because it will provide mechanisms for extra rigidity in some parts of the plant. We're going to find out that when water moves through the entire plant body, they move through systems of plant cells that are just hollow. They're dead. And that's really good, efficient movement of water. There's no, cytoplasm to get in the way. It's just like a straw, when you suck water through a straw. So it can be very useful, even though it sounds weird that they deliberately kill off some of their cells. It's useful. All right, so the primary cell wall, secondary cell wall. So just be aware that this artist, either they're thinking that the cell hasn't quite got the signal to do programmed cell death, or they miss the fact that the secondary cell wall leads to an empty cell, a dead cell inside, because you can see they drew the primary cell wall, secondary cell wall, and there's still all these organelles. So just be careful about that. Sometimes, more than once, I've seen textbooks where I can tell the artist and the person who wrote the book might not be communicating too well with each other, and they make some mistakes in the drawing. So often the artists are not experts in that field of study. They're really good artists and illustrators, but they might not have the expertise. But anyways, so here, a little bit more going on here with the image here, where you have these 4 neighboring cells, and they've drawn in the nucleus again on each. For some reason, they really like purple for that. And then you can see the vacuole in each cell being larger than anything else. And then they've drawn in some of this greenish stuff is the part of the membrane material that actually flows and kind of forms it's a membrane lined tunnel that leads from one cell into the next. And it is allowing, these tunnels allow for transport of stuff from one neighbor cell to the next. The fancy term, the plural term is up high there, plasmodesmata. I want you to know that term. So the plasmodesmata, that's plural. Plasmodesma is singular. So the plasmodesmata is the fancy term for the tunnels that connect neighboring cells. So they're trying to show that they're lined with this membrane that extends from the, first cell to the second cell. That's accurate. Okay. And so it does allow, it can't, the way the artist drew this, it's very easy to see that the nucleus certainly can't go through. But small chemicals, small proteins, small enzymes, hormones, other secondary metabolites, some small chemicals can fit through there. So it's a way to communicate and pass molecules between neighboring cells. The image in the lower right corner is an electron microscope picture of plasmodesmata from one cell to the next. And it depends on what part of the plant, like what plant tissue you're looking at. Some have lots of plasmodesmata connections, some are modest to few. So it just depends on how much they need to communicate with each other, how many plasmodesmata they might have. Here's another image trying to show the plasmodesmata connections. So those are neighboring cells that are connected through these tunnels. And then here is a more full rendition of a plant cell with more of its organelles being shown. And then you can see at least one of the one plasmodesma here. So the way the artist drew this, they were probably thinking this is still not a fully mature plant cell. And the reason I'm saying that is that the vacuole is so small. I mean, it starts off small, but by the time that plant cell is fully mature, it'll look more like this, where you have a big vacuole. Okay. And I'm making a big deal about this because the vacuole, you want it to get big. You want a mature plant cell to have a big vacuole. That is another reason or another way for plants to fight gravity, believe it or not, because this big vacuole it takes up so much room in the cell, and then it's jammed, hopefully jammed full of water. And if that's the case, that is a lot of pressure, water pressure. And that water pressure pushes against the membrane of the vacuole. Then the vacuole pushes against all the other contents of the cell, which then push against the cell membrane, which then push against the cell wall. So all that water or turgor pressure then allows for that part, those cells, to be nice and rigid. I'm sure you've bought or eaten lettuce that's all wilty. Not much fun. I brought some can't remember what kind of lettuce, lettuce you put on a sandwich. I brought it home from the supermarket, forgot to store it correctly, and I just stuffed it in the fridge because I was in a hurry. Took it out the next day, it was all wilty. Ever have that happen? You're like, oh man. If that happens, just put it into a bowl of water. So you might want to cut some of the tips of the leaves. So you've got some cut areas. Just submerge it. Some people say hot water. I do cold water. And just leave it there for like 30 minutes and it'll just plump right up. What you're doing is you're allowing the plant to have the necessary access to water and time to, again, get its vacuoles full of water. And then the leaf, the lettuce leaf will start to get looking more fresh and not so wilty. It's a good trick. So if you have a wilty lettuce, what happens is that the water has come out of the vacuole. And so then it's not able to stay nice and rigid. It can't fight gravity. And it's not much fun to put on your sandwich either. So you might have noticed that maybe when in BI 111, when you did some microscope work, I know that you have looked at plant cells. I remember the first time I was looking at plant cells and I'm like, why are the chloroplasts all smushed off to the side? And it looks empty in the middle. And I was so confused by that. And it wasn't until the plant class that I learned when you look at it under the microscope without any special staining, you can't see the vacuole. You can't see it. And so you'll see all this stuff around being squished off to the side and it looks empty. It's not empty. You have to do a very sophisticated staining technique to stain the membrane of the vacuole. And that's why you just don't see it. So if you happen to look at cells on the microscope and everything's jammed around the perimeter, that usually means it's a very healthy cell that has a nice vacuole and there's lots of water in there. Here's another example where they're showing the vacuole being the largest. So if you have a nice mature cell with a nice big vacuole, that the number one ingredient inside any vacuole in a plant is water. And so when the water is in there and it's pushing against the membrane of the vacuole, which pushes on against everything else around it, then you have a nice turgor pressure developing, or water pressure. And we're going to talk a lot about turgor pressure in this class. So I wanted to introduce that now. There are other things inside the vacuole too, not just water. Water is the number one ingredient, but there can be proteins and enzymes in there. There can be pigments. Some of the pigments that you see, like cabbage leaves, the purpley pigment, it's actually stuffed in the vacuole. Not all pigments are there. It depends what plants you're thinking of. So sometimes they'll stuff some chemicals that they don't want circulating in their cytoplasm. Not the best for them. They'll sequester it into their vacuole. So at least it's keeping the nucleus and the chloroplast and the mitochondria safe from harm. So sometimes they'll use the vacuole as kind of a quarantine area as well. If you've ever don't you hate it when you have an apple and then you accidentally drop it, and if it hits like a corner and then it gets all brown and mushy? And what you've done accidentally is you've ruptured the vacuoles of that piece of the apple. And some of the enzymes that are in here are enzymes that break down cell components. And so the enzymes are freed and they just start going nuts and start breaking down the cell membrane and organelles, and then it gets all mushy and all that region dies. So the vacuole has some important roles. It's not just turgor pressure, but turgor pressure is huge. That is a really important role, because if they're nice, turgid is the word. If those cells are nicely turgid, that tissue is nice and upright. It can fight gravity. It's very healthy. Okay, so another thing that's unique to plants that we don't have, unfortunately, are chloroplasts. And so we're going to be talking a lot about chloroplasts in this class when we get into photosynthesis. A couple of things to think about right now is the basic anatomy. So here you can see that there are actually two membranes that surround the internal components of the chloroplast. So there's an outer membrane, which this is green, we're putting it, then that orange thing, that's called the inner membrane. But then you also see this weird funky stuff in the middle. These green areas that come stacked up, and some are like ridge parts. That's what I call the internal membrane. Okay, internal membrane. Here's another depiction of that. So you can see the outer membrane, the inner membrane, and then that all of this in the middle. Again, that's what I call the internal membrane, the fancy word for that. Okay, so I'm going to take issue with how the artist drew this. So when you look at the entirety of that internal membrane, you see like the stacked regions that look like coins stacked up, and then you see like bridges of membrane in between those stacks. That in its entirety, all of it, is actually called the thylakoid. Okay, so the thylakoid is actually all of this. Okay, all of that internal membrane. Then they further like dissect what's going on here. So the stacked regions, just the parts that look like coins stacked on each other, the stacked regions, those are called granum. If you talk about just one, it's granum. If you talk about more than one, you call it grana, Grana. So if I say, oh, look at all the grana, what I'm referring to is all these stacked regions of the membrane. So that's all membrane material. It's all kind of stacked up like that. Then if I say, oh, look at the bridges between the grana, the fancy word for that or phrase for that is stroma lamella. And then if you take it like all the bridges, then you add an E on the end of that word, and it's stroma lamellae. Put an E on it. So when I go, oh, look at all the stroma lamellae, what I'm thinking about are all the relatively unstacked regions where they look like bridges of membrane. So there's more to it. And this picture, the way the artist labeled it, I disagree. And so do every other plant physiologist. So the entire internal membrane is called the thylakoid. Stacked regions of that membrane are called grana. The bridge-like regions are called stroma lamellae. Now, the reason they had to be so nitpicky and give all these different parts different names is that different things go on in these different parts. And so they had to have a way to denote exactly what portion of the membrane they're talking about. So we'll get more into what exactly is going on, what's going on in the grana versus stroma lamellae. Yeah, there's some different things. It all has to do with photosynthesis, but we'll get into that later when we start covering. But for right now and for the first exam, I do want you to know the anatomy of the chloroplast. That's a very basic concept that I want you to learn now rather than later. And of course, depending on the part of the plant you're looking at, they can have a lot of chloroplasts in that cell. So of course, leaves would have a ton of chloroplasts in their cells. Other parts of the plant, they might not have any. If you were a plant, can you think of a part of your plant body you wouldn't really devote any resources to making chloroplasts? Roots. Yeah, right? If you bury your roots in the soil, why would you put chloroplasts in there? Chloroplasts are like photosynthesis, light. So if you're building your root tissue, yeah, don't put chloroplasts in there. Okay. You can do other plastids. There's different, like chloroplast is a type of plastid. There's other plastids, like if you took Plant Kingdom, you learned about amyloplasts, which are plastids in the root that store starch. And that makes a lot of sense because roots are very good at storing up carbohydrates, like sugars, complex carbohydrates. That lower image, figure D, you see ATP is getting made, and they're trying to draw a proton gradient. So we're going to be talking a lot about a proton gradient, which there's different ways to describe proton gradient. I can't draw a proton. My pen is not happy with proton gradient. So why am I saying gradient? Well, if you look inside here, you see lots and lots of protons, and then outside there's hardly any protons, and what's separating them is a membrane. So this is actually the thylakoid membrane. What they did is they're peeking inside. It's like they cut open one of the granum, and they're looking inside of the membrane. And oh yeah, okay, there's the word. Looking for the word. So the lumen, I want you to know that term too, please. Okay, my computer is really not. So the lumen is if you were to crack open any part of the thylakoid, really, it doesn't matter what part of the thylakoid, you could crack open the granum area of the stroma lamellae. But if you're inside that membrane, we call it the lumen. Just like the inside of your intestines, that's referred to as the lumen of your intestine. Okay. And then the outside portion of it, outside of the membrane, all the watery stuff that's floating around is the stroma. Okay, the stroma. I need you to know those terms too, because when we get into photosynthesis, we're going to find out that certain things, really important things, happen in the lumen, and other things happen in the stroma of the chloroplast. One huge concept is this idea that they've drawn of a proton gradient being used. So the gradient across that membrane there, once you establish a proton gradient, it sets up the situation where then you can synthesize ATP. Any questions on the chloroplast? Wouldn't it be awesome if we had that? We could just go to the beach every day, lay out in the sun, like, I don't know, 20 minutes. Well, true. In this area, it is cloudy here a lot, but would it be nice? Yeah. And then we wouldn't I don't know about you, but trying to figure out what I'm going to eat for breakfast, lunch, and dinner is such a pain. It's like, just let me lay out in the sun and then I'll feel full. I'll be fine. I can go about my day. Unfortunately, we can't do that. So, here is an electron microscope picture of a real chloroplast. And so you can see the relatively stacked regions of the thylakoid membrane, the grana, the relatively unstacked region, stroma lamellae, all that kind of grainy gray stuff that the membrane is floating around in. That's the stroma. Kind of hard to see the double membrane on there, but it's there. So very interesting. The other concept just to be aware of and just jog your memory of the mitochondria. So we have mitochondria. Plants also have mitochondria. And again, depending on what part of the plant body, they may have relatively few mitochondria in that cell or a lot, just like you do. In your muscle cells, you tend to have a lot of mitochondria. Some similarities in the sense that there's a double membrane, but it's quite different from the chloroplast, really, because you have this outer membrane, and then the inner membrane is actually invaginated like that to form the so-called cristae. Okay? And then the watery portion inside of all that is called the matrix. So the infolding like that, the imagination of the internal membrane, inner membrane, that's just to provide a lot of surface area to stop a bunch of proteins and enzymes. into the membrane. And so here they're trying to show that idea where you have a lot of activity in those cristae regions. And again, what the artist has drawn, you have lots of protons on one side of the membrane, less on the other, so you got a proton gradient. All right, so that is another kind of analogous thing going on, you know, with what I just was showing you in the chloroplast, and now with the mitochondria. You have this proton gradient across the membrane. Anytime, no matter what organism you're looking at, what cell type, if you see that there's a proton gradient established across the membrane, that should ring some bells for you to look closely at what's happening. Because very often it's being used, that gradient is being used to synthesize ATP. So we'll be talking about that. So this idea that they're not really showing it in these drawings, but I'm hoping that you've learned in basic biology class that mitochondria and chloroplasts, they have their own DNA, which is wacky. And it's circular DNA, which is wacky because that's what microbes have. And eukaryotic organisms don't have that. They have linear. There's a few microbes that have it, but most are circular. And so that led to the idea posed by Lynn Margulis that the chloroplast and the mitochondria were ancient bacteria that were engulfed by larger cells during the evolutionary process and led to them persisting inside that bigger cell. Instead of being eaten up by the bigger cell, they persisted and morphed into, you know, evolved into what we now call the chloroplast and mitochondria. And when that was first proposed by Lynn Margulis, she actually did this for her PhD thesis. And when she proposed that, they just didn't know. Everyone's like, no, you're wrong. And this is back in the 60s, where being a female scientist was quite tough. And so it took a lot of effort. She had to do a lot of further study on it, and then other scientists got involved. Actually, that was during the Cold War. And other scientists, like over in Russia, were also working on this. But back in the Cold War days, we didn't talk to each other. And so we didn't know till after the Cold War and things warmed up that, oh, there were other people also investigating this and coming to the same conclusion. So now it's probably in your freshman biology textbooks. It should be, at least the ones I've seen there, it's in there. And so very interesting concepts. So the idea, we're not going to focus too much on the mitochondria, but we certainly are going to be looking at the chloroplast in more detail for sure, because that's where the whole concept of photosynthesis is cooking along. All right, before we zoom out of the plant body, let's take a little bit of a look at tissues. So when you have cells neighboring together, you have the middle lamella and pectin, all kind of cementing them into a tissue. What do we got going? Well, we got some different types. There's dermal tissue. We have dermal tissue. That's our surface tissue, our skin tissue. Plants have a surface as well that we call the dermis. And so the very, very surface is the epidermis, just like we have an epidermis. And they have a special addition to their epidermal layer because they cover it with wax, which is called a cuticle. So cuticle is this relatively thin layer of wax. So if you think about it, if you cover your above ground parts with wax for the most part, that's really going to help seal your body and keep you from losing water. And that's really a huge goal of the cuticle, is to seal up cracks and crevices and make sure that any water that leaves the plant body, the plant has control on. And they do have exquisite control on their water leaving their body for the most part. We'll talk more about that. Well, actually, we'll talk about it next. So guard cells. So on the surface of the plant body, if you look at a leaf, you will see that waxy cuticle covering the whole leaf, both top and bottom side. But under the microscope, you'll see some interesting looking cells that we call guard cells. And really, there's two guard cells. Okay, so my hands are guard cells. And then they surround a pore. All right. And then you take that all together, that complex together in the pore with the two guard cells on either side of it, and you call it a stoma. Plural would be stomata. I almost always say the plural, stomata. So when I say stomata, we're thinking about the guard cell and the pore that they guard or surround. And those guard cells are capable of movement to either reveal the pore or close over the pore. And they have really fine-tuned control of their guard cells. Like, how much should I reveal the pore? Should I just reveal the pore 10% open, 50% open, 100% open? And minute by minute by minute, they are making that decision on what they should do based on they're assessing their environment. How much water do I have access to? Am I going to get dehydrated? And so they are constantly assessing that. They don't have the luxury like we do where we can just go to the refrigerator or go to the sink and get some tap water. So I'll show you some pictures of that. And then there's ground tissue. So if you go in another, layer, then we start talking about ground tissue that makes up like the internal parts of the plant body. So you have the ground tissue. And so there's different types of ground tissue. Parenchyma, and I'm sorry, it's a weird spelling. It sounds hard to pronounce, but it's called parenchyma. That's the most abundant type of ground tissue. So when you look through the whole plant body and you look at what kinds of ground tissues there, parenchyma wins, hands down. There's more parenchyma tissue than anywhere else, than anything else. Leaves are kind of a special case. When you look inside of a leaf and look at their ground tissue, we tend to see palisade parenchyma and spongy parenchyma. All right, so these are types of parenchyma. Palisade parenchyma and spongy parenchyma. And we give them different names because visually they look very different. And so we call them different things. But they're a type of parenchyma. Okay, so let's look at a leaf. Here's a leaf, and you can see the artist tried to draw that waxy cuticle over the surface of the epidermis. And then they're saying if you take, cut the leaf cross section and take a chunk and look at it and blow it up a little bit. That's what you're seeing in the main portion of the image here. So palisade parenchyma are the cells that are toward the top of the leaf here. So they look like columns or palisades, and they're kind of smushed right next to each other. So that's the palisade parenchyma. Some people call it the mesophyll cells. I tend to stick with parenchyma. It's a type of parenchyma. Mesophyll is an extra word that you have to try to link into parenchyma. It's just kind of a mess. So I try to stick with these are parenchyma cells, palisade parenchyma. And then the spongy parenchyma, you can see are more round and they're not so smushed together with each other. And in fact, there's quite a few air gaps or air spaces in between them. That's by design. That's not an accident. Okay, so when plants are designing their leaves as they're growing, they do leave some internal air spaces. If you imagine, we had some windy days. If you imagine if this was just a solid chunk of palisade and spongy mesophyll all jammed together on a really windy day, those leaves wouldn't be able to really bend with the wind and let the wind, roll off it, so to speak. And so it would rip the leaves off the trees. They've done some nice slow videography of putting leaves in a wind tunnel. And then you can see how the leaves actually kind of roll with the wind rushing across it. And so those air gaps help that flexibility. So that's an important reason. But we're going to find too, there's another really important reason for these air gaps. And it has to do with how they move water through their leaves and out the stomata. So here you can see the bottom of the leaf. You see these weird looking guard cells. They often they look like the shape of a kidney bean, like a red kidney bean. They kind of look like that. And then there's the pore in the middle there. So you can imagine those guard cells can flex and bend away from each other, or kind of, like praying hands come close to each other. And they can really regulate the size of the pore. So the stomata are really important. And that's, when the water is moving through the plant body, if those stomata are open, you'll get water loss for sure. But if you have your stomata open, you're also able to get things in, which is good, right? Because you want CO2 in so you can make your sugars. But as soon as you open your stomata, you automatically have water exiting your body. You can't help it. It's not like you can say, oh, water vapor, stay in. CO2, come in. You can't do that. By the laws of physics, it doesn't work. So as soon as they open their stomata, water starts exiting as water vapor, but they get the CO2 in. And so now you can appreciate how much plants have to be aware of their surroundings. I gotta figure out how much water I have. Is it safe for me to open my stomata? I want to do photosynthesis. I need that CO2 coming in. I have to open my stomata. Oh, but I'm not feeling much water around me. Uh-oh. So you can imagine there's going to be cases like that, often on a daily basis, depending on where the plant lives, where they're in a conundrum of, what do I do? And so we'll talk about that in a little bit more detail as we start getting into water movement, and then of course photosynthesis. But let's stop there, and make sure you go to lab tomorrow. And then in Friday's lecture, we'll get into a few more things about anatomy, and then we'll start talking about water and water movement. Okay. With the CAM plants, you mean? Yeah, the CAM plants. Yes, there are just a few CAM plants that can actually turn off the CAM pathway and just be more normal.